Okay, let's move on to the next topic. The next topic is going to be microscopy and magnification. So there are two parts to this. There's the theory part of it, which is kind of like the actual microscope itself, light microscopes, electron microscopes. And then we have the second part of it, which is a calculation question. And 9 out of 10 exams, they will ask you a calculation question on magnification. So it's good to point this out. But let's look at the theory part first. First part will be the light microscopes and the electron microscopes. So I believe the ones that you use at school are light microscopes. You need to know that they were light microscopes. They were invented first. They have a maximum resolution of 200 nanometers. And knowing the structure on the side, you can see a diagram. You need to know each of those individual parts of the microscope. So the eyepiece lens, it has a 10 times magnification. So just from looking into the eyepiece lens, that will already give you magnification. Then you have 3 objective lenses with which magnify even more, and that's times 5, times 10, times 25. I don't think you need to know numbers too specifically, but that's just extra information. The electron microscope, they were invented a lot later. And instead of using light, so light microscope, it uses light, as in the name. Electron microscopes, they use a beam of electrons instead. And this means using a beam of electrons, it has a much higher magnification and resolving power. So resolving power meaning resolution. This allows us to have an increased understanding of subcellular structures. So when a question asks you about these microscopes, sometimes they do ask you to compare them. You just need to know light microscopes uses light, electron microscopes uses beam of electrons, and electron microscopes have a much higher magnification and a much higher resolution. You need to mention both, not just magnification. You need to mention resolution as well. And with that, it's also important to note that electron microscopes are a lot more expensive compared to light microscopes. So that's why the school gets light microscopes instead. On the diagram on the right, we'll just quickly walk through it. Just a few of the things. So the stage, that holds the specimen, so whatever you're looking at. The specimen essentially is on a microscope slide and it's under a cover slip. You also have the coarse focus and the fine focus. The coarse focus usually just moves very quickly to move the stage closer, so you can see the specimen more closely. And the fine focus moves it very, very finely, very, very slowly, so that you can very quick, like slowly adjust to seeing the specimen in a very clear image. Okay, now let's move on to the math part of it. So I'm gonna give you guys about 1 or 2 minutes to do this question. And I will walk through it with you after giving you a minute or 2. Okay, I think you guys probably have had a good look at the question. Let's walk through it. So the biggest mistake that students make when they're doing magnification calculations is the units. This is because the calculation itself is actually quite self-explanatory. It's just one equation and you just need to rearrange it. The biggest mistake is not looking at the units very carefully. So let's just have a look at this question. Okay. Right. When you're doing these magnification calculations, you already get a mark for just writing the equation out. So I just recommend writing the equation. So this would be, in this case, image size over actual size gives you the magnification. So I over A equals M, or however you want to memorize it. Rearranging the actual equation also gives you another mark. So just by doing that, you're going to get 2 marks in the question already. So it's important to you write the equation and then you rearrange it. So don't be lazy and just put in the numbers, because sometimes they might not give you marks for that. They usually do, but it's just safer to write this down. Then you're going to put in the values. So in this question, it tells you that the length of the cell that you're seeing is 4.8 centimeters, and the magnification is 400. So you just substitute the values, rearrange it, and you see that actual size equals image size over magnification. So 4.8 over 400, that gives you 0.012 centimeters. Now, here in the question, it is asking to give your answer in micrometers. So you have to be able to convert centimeters to micrometers, which in this case is 120. So just have to emphasize this again. It is so extremely important that you write down the correct units. So make sure you're converting them correctly and making sure that you're actually reading the question properly. So be very, very careful. But this is just how you break down the question. If you have trouble with math questions generally, because there's a lot of math and sciences in general, especially physics, do as many as you can and try to remember as many of the equations as you can. So with this question, there's not much I can really break down other than be careful with units and do more math questions. Okay, let's move on.